Comments? You know what the title of it is? Summarize it in one word. Empathy. Okay? I show that because you're all going into healthcare in some form or another, and you always have to be on guard with yourself because you don't really know what's going on with the people that are all around you when you're in a healthcare situation. A lot of things are happy, they're really happy. A lot of them are really sad. And some of them are just downright disgusting. They're so sad. But it's all part of life. And I like to show that as much for you to think about in your job someday. But I also want you to think about it here today. We're going to take this class as a group, as you've already figured out, and we're going to work together, and we're going to help each other. And the only way, I believe, that anyone will be successful is if we're all successful. And we have to help each other. And just remember that there's people in this room that are going through all of the things that you just saw right there. So let's be nice to each other. Be nice to me too, would you? This is my son with his first baby. That baby is now 7 years old or 8 years old, and she's already been in a couple of community theater roles as a little actress, and she's pretty good, I have to tell you. So we're going to start talking about the brain, the spine, and the cranial nerves. Now, just so you all know, if you remember back to 231, the last two chapters of 231, one of them was entitled The Histology, Cellular Structure of the Central Nervous System. And we learned about ependymal cells, and we learned about the different nerve cells and Schwann cells and oligodendrocytes and all that stuff. You still got to know that. Because we're gonna hit on those words as we go through these next few chapters. So don't think you can just say, "Oh, well, I've already tested on chapter, 12 and 13. I don't have to worry about them anymore." It doesn't. You can't really disconnect them. So just keep that in mind. And I would recommend that you take a little bit of time to at least look at all the pictures. If you don't want to read it, I know reading's hard. At least go look at the pictures and flip through that, because I don't want you to let that screw you up. I can't completely separate them. Chapter 13 was the spinal cord, remember? And we learned about the spinal cord, and you need to review that too. Once again, we're just going to assume that you know it. But it would probably help you to review it. And if you want, you can go back and on the list of lectures, you can actually even sit and watch me lecture on them like I did if you were in my 231 class at the end. Okay. That's what I just said. The human brain. You don't have to know any of these numbers. You don't have to know how much it weighs, how big it is. It's probably reasonable to know that it's about 97% of all of the body's neural tissue. So almost all of your thinking activity and your brain activity goes on right here. The only other 3% is that spinal cord. Well, the spinal cord is part of the central nervous system, but the nerves that go out to your arms and legs and all that jazz. The average brain weighs about 3 pounds. Albert Einstein's brain was not any bigger than anybody else's. He just used his more than most of us use ours. There's 6 regions of the brain, and I'll just tell you, you got to know these. That's just part of the deal. There's the cerebrum. This is from the top to the bottom. The cerebrum is the part that thinks. It's your gray matter up here on top. Your cerebellum is the part of your brain back here at the lower. It controls all your movements and so forth. Keeps you coordinated. Diencephalon is kind of the midbrain, as is the mesencephalon. The pons is a part of the brain that kind of connects the spinal cord up to the cerebellum. And then the lowest part of the brain is the medulla oblongata, which is the brain stem. Some people call these things part of the brain stem as well. But the medulla is the brain stem. It doesn't really do any thinking, but it's quite important in how it makes your heart beat. It makes your lungs breathe. It takes care of a lot of things that happen in your body that you don't want to have to think about. And I would just say that not one of you has thought in the last 2 minutes about, well, I think it's time for my stomach to churn again. Not one of you has thought, I think I need to speed up my heart rate a little bit more. I'm getting kind of nervous about this A&P stuff. All that stuff just happens automatically, and it all basically happens in the medulla. So this is going to run through these things several times. I find this kind of frustrating sometimes in these slide sets, but sometimes repetition's not all that bad. The cerebrum is the largest part of your brain. It's the top part. It's the part that separates us humans from most lower animal forms. We have a lot more cerebrum to think about things, and allows us the higher function. Allows us to dream, allows us to have memories, allows us to put our words together, to talk, and so forth. There's a right and left cerebral hemisphere. You've heard about somebody, oh, he's so right-brained, or she's so left-brained, and all that stuff. We'll talk about that a little bit more. It's not really very clear-cut as to what's happened on the right and the left, because basically both sides are involved in all of those functions. The surface layer of the cerebrum on top is gray matter. What is it that makes gray matter gray and white matter white? Chrissy, what makes gray matter gray? I'm sorry. Yeah, go ahead.
Isn't it that the gray matter doesn't have
Robert. Dan. Come on. What is it that makes it dark? Missile bodies, cell bodies, the nucleus of the brain cells, right? That has the missile bodies, has the nuclei, has the nucleolus, all that stuff. That's what makes it dark. So when you're talking gray matter, you're talking about an area that has a lot of cell bodies in it. When you're talking white matter, you're talking just fibers, thousands and thousands and tens of thousands of nerve fibers, axons, all going in the same direction, carrying the same sort of information, and they're usually always myelinated. What does myelin do for us? Insulation. To speed up the signal. Yeah. Okay? So that's the basic difference between white matter and gray matter. Just remember that on our brain on top, that the gray matter is on the outside, whereas on the spinal cord, the white matter was on the outside. Remember that spinal cord? Remember that thing that looked kind of like this? And it had these areas that kind of looked like that. This is the gray matter in here, and this is the white matter out there. So it's just the opposite when you get up top. Okay? We'll have brains for you that you can cut open and look at, and they're not human. There's a few old 231 suits, I think, should be there. We refer to the cerebrum oftentimes as the neural cortex or the cerebral cortex. It's the part that really does the thinking. And so when these football players keep getting knocked on the head and get knocked unconscious, they're suffering little bleeds and little bruises in their cerebrum. And that's why they don't think real good when they get older. And they're starting to die from it too. The surface of the brain, of course, has got ridges, which we call gyri, and depressions, which we call sulci. I'll show you some pictures here in just a minute. And then it has a couple of deep grooves that we'll learn about as well. The cerebellum is the second largest part of the brain. It lives back in the back of your head. It coordinates repetitive body movements. So I'll bet that most of you have seen people with cerebellar ataxia, and they want to move, but they just can't control their muscles, and it's very difficult for them. That's because they can't coordinate all these body movements. And There's two hemispheres of that. They are covered with a little bit of cerebellar cortex over them. We'll show you pictures here in just a minute. The diencephalon is kind of the midbrain. It has 3 parts here. I don't know why they do this. I don't know why they say it's in 3 divisions: the left thalamus, the right thalamus, and the hypothalamus. I just think you ought to think of it as the thalamus and the hypothalamus. Now, what do you remember about the hypothalamus? What? It's at the bottom? Yeah. What does it do? You remember? Temperature regulation, that's right. Remember when we talked about temperature regulation in 231, and when you get hotter, your blood gets hotter, your blood, as it flows through your brain, it flows through your hypothalamus, and your hypothalamus reads your temperature and decides whether you need to sweat, whether you need to shiver, or do all these other things all these controls that we have to control our temperature. That's the hypothalamus. The thalamus, if I could describe the thalamus to you in one sentence, I would say the thalamus is a layover. It's a wayover point for signals on the way from your body to your cerebrum. In other words, if you step on a nail with your foot, that is sensed by the nerves in the skin of your foot, and it's carried up to your spinal cord, where it usually crosses over from one side to the other, and then it synapses with another nerve that carries that information on up to your thalamus. The thalamus is where almost all the sensory input to your body has a way station, has a connection, and then the thalamus kind of decides, well, is that really important enough to send on up to the brain? Because if we send it up to the brain's going to have to think about it, because that's what the brain does, the upper brain, it thinks. And do we really want to burden it with that information or not? So the thalamus kind of weeds out a lot of signals that are going up to the brain. And I probably used this example with you before, but how many of you have been sitting here while I've been talking and thinking, man, I can hear that fan and that projector up there. Have you been noticing that? Not until I said something. Yeah. So, but the point is, you've been hearing it all along. We get all of this information into our body all the time, into our central nervous system. And the question is, how much of it makes it up to the upper parts of our brain where it actually gives us something to think about. Now, I can tell you that if that light blew out right now, we'd probably all notice that the fan is still blowing, maybe. That would bring our attention to it. But so much input that comes into our body all the time is kind of weeded out. And the thalamus tends to be where that happens. One other example. Always cracks me up when I ask that question about the fan. 10 people say, yeah, I've been listening to that fan. That bugs me, that fan. I never hear it. Anyway, here's the next question. How many of you have noticed in the last few minutes that your shirt or your blouse is touching your right shoulder? How many of you have noticed that? You see, but you've been feeling it all along. It just hasn't been going all the way up to your cortex for you to make it to your brain, to reach your conscious level. Sessions are getting weird. Okay, so the thalamus relays and processes sensory information. That's what we've been talking about. The hypothalamus, we haven't gotten to the endocrine system yet, but the hypothalamus works very much in conjunction with your pituitary gland and oxytocin and all these other hormones that work on your emotions and autonomic functions. What are autonomic functions? Automatic functions is how I always describe them. It's things like heart rate, blood pressure, respiratory rate, and so forth. the stuff we need to keep happening in order to keep living, especially to make it through the night. Pituitary gland is really kind of part of the hypothalamus. It's connected to the hypothalamus via the infundibulum. We're going to come to that in about 3 more weeks in detail, and then I think it'll make a great deal more sense. The really important part of this hypothalamus and pituitary gland is that is the place where our endocrine system, our hormone system, butts up against our nervous system, and they tell each other what to do, kind of at the hypothalamus. The brainstem, lower down, includes the mesencephalon, the pons, and the medulla. I don't like to beat you up about this. I don't think these are all that important. I don't want to say they're not important. You need to be able to name them. You need to be able to identify them on pictures and on the sections in lab. But let's just face it, these are pretty simple things. The midbrain, the mesencephalon, mesen means midbrain, processes sight, sound, and some reflexes. It keeps you conscious. If you think about consciousness, if someone is conscious, they have pretty good function of their brain. I mean, that's a huge deal when you lose consciousness. You know, that bugs me. Yeah. Well, I jumped ahead a few, didn't I? Yeah, here's where I was. Okay. Midbrain maintains consciousness. If you ever worked 911 and somebody calls you about somebody that they think's had a stroke or has had some kind of problem with their heart or anything, the first question they're going to be asking you over the phone is he conscious? And when you say conscious, what does that really mean? Well, that means they're awake, right? It means they can probably talk, they can probably see, they can probably hear if you're yelling at them. So that's consciousness. And that is if you can do that, most of your brain's working pretty good. Okay? I think that's a thing to keep in mind. The pons, I like to think of the pons as nothing but the connection between the spinal cord and the cerebellum, the one that maintains your coordination. Connects the cerebellum to the brainstem. Medulla is the lower part. I remember all the old Daffy Duck commercial and Daffy Duck comics. He used to always talk about the medulla oblongata and Walla Walla, Washington. So that just comes back from my childhood. But anyway, medulla is the lower part of the brainstem. It regulates the autonomic functions like heart rate, blood pressure, digestion. I always say that's why the guys from the mafia, when they want to shoot somebody and kill somebody, they always shoot them right in the back of the head right here because they want to go through the medulla. If you go through the medulla, their heart doesn't beat, they don't breathe. They're pretty dead. That's one thing you can learn in A&P is how to kill someone. It's an important point. All right, so here's a picture, and I could have started with this, but just looking at it kind of generally here. Here's the cerebrum. It's all of this up here. That's the upper brain, the gray matter of the brain. Here's the cerebellum, the lower part of the brain. This down here is the spinal cord, and so in between here and extending up inside the brain itself is this part, which is the medulla oblongata down here at the very bottom, just above the spinal cord. The pons, which is this kind of knobby thing that connects across, it basically connects this cerebellum to the spinal cord, which is where all that information comes up with muscle, with sensory and motor input. And then the mesencephalon, that midbrain that we talked about, is up in there. The diencephalon above that is the thalamus and the hypothalamus, which we've already mentioned. And so I'm going to just kind of leave that at that. Let's talk about embryologic development for a minute. When we start developing, it's like one sperm cell hooks up with one egg cell, and we call that a zygote. Very good. You remember 112. Yeah, that's a zygote. It's a fertilized egg. It splits from 1 to 2 to 4 to 8 to 16 to 32 to 64, right? And all the cells look the same, right, at first. But then they start differentiating. And some of them turn into brain cells, okay? And the way the brain is formed, it's formed of what's called the ectoderm. We'll come back to this in the last chapter of A&P when we talk about embryology. The way we do it, the reason we do it that way is until you know what all the organs are and how they all fit together, it's kind of hard to understand how they develop. So if you go back then and you say, well, this is how these things develop, then it sort of makes sense. But anyway, the brain becomes a sheet, and then it has to fold over. And as it folds over, it then creates this thing called the neural tube, which is all the neural tissue surrounding that empty space in the middle. And down in the spinal cord, we call that space in the middle the central canal. And up in the brain, where the brain folds around, we call those empty spaces the ventricles. That's right. Good. Now, if things don't fold all the way around, let's say they kind of go around, but they don't go completely, that can be as minor as what's called a spina bifida, okay? As it gets worse and worse and worse, and the brain just really doesn't even connect around, we call that anencephaly, and that's, anencephaly means no brain. And babies that are born with anencephaly, they live 2 days, 3 days. It's really a tragedy. If you want a really depressing evening, go home and read about the support group for parents of anencephalic children. And it talks about how they found out their kid was anencephalic, usually from an ultrasound, and then how they went through all these discussions, decided what to do, and most of them then go ahead and have the child delivered, and it lives a day or so and dies. I'll stop and tell a story. I only saw one anencephalic kid in my medical career. I was standing in the emergency room at Bethesda one afternoon, and Marion Jordan came up to me and said, "Sessions, what you doing this afternoon?" I said, "Not much. I'm on ICU duty this month. I'm covering the ICU, but we only have like 3 patients in there, and I've got a couple of interns that could look after them." He says, "You want to go harvest some kidneys with me?" I said, "Yeah, sure, let's go." He said, "Well, run upstairs, get your hat, because you have to have a military hat, although we doctors never wore one." So I went up and got my hat, and he said, "Meet me back in the ER." And so I came back, and there in the ER, there was, 2 nurses, 1 surgical nurse, 1 transplant nurse. There's 3 or 4 OR techs, instrument technicians, and so forth. And there was me and him. And about that time, this ambulance pulls in, and we all threw they had a bunch of boxes of equipment. We threw all that into the ambulance and took off across Washington, D.C. to Andrews Air Force Base. Same place the Pope is landing this afternoon, by the way. I heard on the news this morning. So we go out onto the runway at Andrews, and there's looks like about a 20-man plane sitting there. It was a turboprop. It wasn't a pure jet. And it was already had the props were turning. The gate was down. We threw all this equipment up into the back of the plane. We all crawled up in the back of the plane, and within a minute we were in the air. It was great. I said, "Marion, where are we going?" He says, "We're going to Norfolk, Virginia." I said, "Cool." There's a big Navy base in Norfolk, big Navy hospital down there. So we landed in Norfolk, and there was another ambulance waiting there on the runway for us, and we all came down, loaded everything off into this great big ambulance, and headed off across town, lights and sirens, went right through. It was so cool to go through places where you paid on the freeway. You know, you're supposed to throw your money in the little thing, and then the gate opens. No way. Boom! We went right through. There was always an empty slot over there on the far right, I think. And we just blazed it through there. I'm surprised we didn't kill anybody. Anyway, we get to the hospital, and the crew is there, and me and Marion went up to the ICU to make sure all the papers were signed, because doing transplant harvests is a very difficult thing. First of all, in this country, in order to remove anybody's organs, the person has to be declared dead by one set of doctors that is taking care of the patient. And in this case, that was the pediatricians who were taking care of this little one-day-old anencephalic baby. The parents knew the baby was only going to live a few more hours, and they had donated the kid's kidneys to be transplanted. And so they checked all the paperwork, and we went up to the operating room and washed up, and you do the operation. It's a midline incision. You go in, you take out the kidney, you take out the renal artery, as much renal artery as you can get, as much renal vein as you can get, and you take out the entire ureter right down to the bladder. So you've got all of it. Did that on both sides. They went into a bucket of iced saline. They had box lunches for us. I'll never forget. It was so nice. Here's your kidneys, and here's a box lunch for everybody in the ambulance. That was very nice. Went back screaming across town, got in the plane, and took off again. And I said to Marion, Marion, where are we going? He says, well, we're not sure yet. He said, we think we're going somewhere up in New England. And but he said they're still doing type testing, tissue type testing on the kidneys and on the baby's tissues, because you obviously want it to match whoever you're putting it into. And so we landed at LaGuardia Airport in New York City. It was snowing in LaGuardia, and we came to a stop, and up comes an ambulance with all the lights and sirens going, and the guy gets out, and he says, hey, is this the kidneys from Virginia? And we said, yeah. He says, okay, I'm getting them. He signed a piece of paper, and we gave him the kidneys, and we took right back off and went back to Bethesda. And I was at home in bed by 9 o'clock at night. This whole thing started at 1 in the afternoon. And the really good thing was the next morning I ran into Marion in the hospital again. I said, Marion, did you hear anything about the kidneys? He said, yeah. They transplanted each kidney into 2 babies, and they're all making urine. The kidneys are working, and they're making urine. So the tragedy of the anencephalic baby saved 2 other kids, and that's kind of a cool thing. But the tragedy of anencephaly, read about it in your book. It's a really tough deal. That was my only experience with it, and I'm glad I can say that. So anyway, let's talk about how the brain, it starts out as a flat sheet of ectoderm, and it folds around and forms this neural tube. And as it does, it leaves this empty spot in the middle. And that empty spot then matures into the brain vesicles, and the ventricles are what become the empty water space in the middle. The book goes to a great deal of energy telling you about the prosencephalon, mesencephalon, and rhombencephalon, the different parts of the brain. Basically, it's the upper brain, the middle brain, and the lower brain. I just don't think you need to know this, so don't worry with that. You don't need to know those words. And here's another slide that's in your book that you don't need to know. So here you have prosencephalon, mesencephalon, rhombencephalon, and then as it continues to mature, this turns into the telencephalon, the diencephalon, the mesencephalon, the metencephalon, the myelin. Okay, so what? Just know that this is the upper brain. This is the part that really does your thinking for you, and these are the lower parts of the brain as they come on down. Don't worry about those names. I just don't think they're important. You can notice over here that towards the bottom you're down into the cerebellum and the pons, and at the very bottom you're at the medulla. But we've already said that. That's all you need to know. the ventricles of the brain are these water-filled things in the brain. You have big ventricles out on either side in the cerebrum up top. They become the right and the left lateral ventricles, one on the right side, one on the left side. Remember, all of the ventricles, the central canal and the spinal cord are all lined by ependymal cells. Remember that? These are these big water cavities on either side. You've probably heard of kids that have hydrocephalus. Hydrocephalus is oftentimes referred to as water on the brain. And basically what happens is these are these ventricles up in the brain that fill with water and they can't empty. There's something wrong. Either they're making too much water or it's blocked and it can't get out. Something down that line, and their head swells up really big. Back in the day, that was 100% fatal. And the stories I heard were always so awful because these kids, their heads would just get bigger and bigger, and you could shine a light on them and shine all the way through because it was all water. And then one day the nurse would go in and the balloon had popped, and the kid's brain had just popped like a balloon. It was all in the bed. But nowadays, we can fix it. And how do we fix it? A shunt. That's right. You put a little plastic tube into the ventricle up here in the brain, bring it out through the skull, out through underneath the skin. You pass it under the skin, down here to the neck, make a little incision, and pass it again down here to the top of the tummy here, under the skin once again, and then you poke it into the abdomen, and you just let it drain into the abdomen. So that fluid is continually coming out of the head, going down through the tube, and dumping into the abdomen, where it just is absorbed by the peritoneal cavity. After these two first ventricles, and I'm a terrible artist and I very seldom ever draw pictures because I'm not very good at it. So if you have the right and the left ventricles here, then they come together and they form the third ventricle. How's that the third ventricle? Well, that's because this is the first ventricle. The one on the other side is the second ventricle. This is now the third ventricle. Yeah, right. So, anyway, and so then back here behind the third ventricle, there is a fourth ventricle. It opens up a little bit, and then it eventually is going to continue on down as the central canal of the spinal cord. As it goes here into the third and fourth ventricle, there's some little holes on the sides here that allow the fluid to come out of the inside of the brain, and then they're now around the brain, and they fill that space, which is called that space that you stick a needle into when you're doing a spinal tap, which is called the subarachnoid space. That's right. And it percolates around here in the subarachnoid space. It also sort of percolates completely around the brain. And up there it's called the subdural space. You've probably heard of people getting a subdural hematoma when they bleed into their subdural space. So that's kind of the circulation of the CSF. This last part is called the cerebral aqueduct. I think I have better pictures here. Yeah, see, this is why I don't draw pictures, because they always have such nice ones. So here's the first and second ventricles, these two big ones that come all the way around and stick out into this frontal lobe of the cerebrum there. And then they come together up here at the interventricular foramen where the two of them hook up, and then they come back into the third ventricle, and then they go down here through this aqueduct of the midbrain, often called the aqueduct of Sylvius. You probably see that in your book somewhere. and then it comes back in here into the fourth ventricle. The way I always remember the fourth ventricle is the fourth ventricle is right in the lower part of the brain. So it kind of touches the pons, the cerebellum, and the medulla are all right around the fourth ventricle. And then the fourth ventricle continues on down here is the central canal, but it also has holes here that allow the water to then go out and around the brain and into the subarachnoid space. Okay. So the brain is a large, delicate mass of tissue containing internal passageways and chambers that are filled with the cerebrospinal fluid. Each of the 6 major brain regions has specific functions. What I've already told you is about as in-depth as I really want you to know it. We'll talk a little bit more about a couple of other things, but you know, I don't really want you to feel like you've got to know everything about every one of these parts of the brain, because it's just, I can tell you in my practice, I never knew it. I thought they got a brain injury, they got a brain injury. You know, that's kind of the way it worked. Usually they were unconscious, and that was about all I had to know. If they're unconscious, they need to go to a neurosurgeon. Send them to Manual. Get a helicopter up here from Manual and get them down the river. So anyway, I try not to whip you up too bad about that stuff. I think it's really important that you know that everything we do, when we think about thinking, every bit of thinking, intelligence, dreaming, all of that stuff, everything takes place up in the cerebrum, up in the top, the neural cortex on top. Let's talk about physical protection of the brain. Obviously, the brain is our most important organ. Without a brain, we're pretty useless. We're at the whimsy of any animal that wants to come along and eat us, or any other human that wants to beat us up. So the brain is very well protected. It's protected by the bones of the cranium. And let's see if you can remember a few of those. Frontal. What? What's this one? Temporal. Parietal. Occipital. Yeah, so they make this box up there that holds our brain in it. Now, the good thing about our brain living in a box is that it's protected from the outside. The really bad thing about our brain living in a box is that if anything happens to our brain, it makes our brain swell even a little bit. It causes an increase in the pressure inside because it can't expand at all. So anything that causes swelling of your brain leads to increased intracranial pressure, which then causes all kinds of problems and kills you, including causing herniation of your brain out of the big foramen magnum, have your brain kind of slop down into your spinal cord. So dealing with high intracranial pressure is really what managing brain trauma is all about. And that's why they'll oftentimes drill holes into the side of the brain just to let the pressure out. They'll give mannitol and other steroids to cause the brain hopefully to shrink down a little bit so it's not so swollen and not under so much pressure, and so forth. Sometimes they even take a big piece of the skull out, stick it in your belly or someplace, and then 3 weeks later, after all the pressure goes away, they build you back together. Yes? That's why it's bad to hold in your sneezes.
That's why I heard it. It like increases the pressure.
Yeah, I think my mother told me that once too. I'm not sure. It does raise your intracranial pressure, but just for a second. I don't think it's too terrible. So, in addition to the bones of the cranium that are a blessing and a problem, you have below that you have the cranial meninges. Those are the layers of protection on your brain. The outer one, which is the really tough one, which is referred to as the tough mother. Dura mater. Durable mother, tough mother. Dura mater is the outside one. The middle one is called the arachnoid mater, arachnoid spidery, like arachnophobia, and it carries blood vessels and so forth. And then the inner one is called the pia mater, and the pia mater is firmly attached to the inside of the brain. And even in our specimens in the lab, you really can't pull the pia off the brain. It's firmly attached to the outside of it. The last thing, let me just mention for a second, is the biochemical isolation of the brain. And what that means is that because our brain is so critical to us, we have a system that occurs at the walls of the arteries around the brain that are very, very specialized, and we call it the blood-brain barrier. And that's just a fancy word for saying it lets in some stuff and it doesn't let in some stuff. So, for example, if you're a diabetic and our normal blood sugar is about 100, and you're a diabetic and you're having a crisis, your blood sugar might be 1200 in your blood they draw for the lab. Okay? But at the same time that it's 1200 all through your body, if you could stick a needle into a blood vessel inside your brain and draw out blood, the blood sugar would be 100. It would be perfectly normal. In other words, these cells, these ependymal cells that work with the astrocytes along the outside of the brain, prevent chemical changes from occurring in the brain. They can keep stuff out and they can keep stuff in order to keep the chemical milieu, so to speak, of your brain normal. Even though it may not be normal in the rest of your body, it can keep it normal inside of your brain. That is called the blood-brain barrier. Now, how do you think we figured that out? Well, that wasn't easy to figure out. One of the ways we figured it out was when you had people that had meningitis back in the day, and you gave them all of these antibiotics that you thought, man, these will just kill that bacteria, no problem at all. But some drugs and some antibiotics could penetrate the blood-brain barrier, and some couldn't. So if you were trying to treat meningitis in the brain, and the drugs you were giving wouldn't go through the blood-brain barrier, you were wasting your time, and it didn't help at all. And it took a long time for physicians to figure out that, wait a minute, some things can get through there and some things can't. And now you can just look on a list and say, these drugs work fine for infections in the brain, and these drugs don't work at all for infections in the brain, because some will go through the blood-brain barrier and some will not. Now, it probably sounds like a little bunch of hocus pocus, me telling you about this blood-brain barrier. Read about it in your book. We'll talk about it a little bit more. We're going to talk, I've got some videos to show you on the cerebral spinal fluid circulation and where it's made, how it moves, and where it goes, and all that good stuff. So we'll come back and start here. I've already said that. Let's skip that slide. We'll start right here. Okay? So take your time, take a little break, go to lab. We don't have a lot to do today in lab. Oh, wait a minute. I'm sorry. We have the safety video for you.
The camera.
Okay, testing 1, 2, yes. All right, this is a second lecture of 232. We actually didn't get too far in the first lecture, so we're starting at about slide 21 or 2 in on the brain, chapter on the brain, beginning of 232. All right. How's it look up there? Does that look good? Maybe I ought to turn the projector on. Thank you. Anybody else need a stapler? All right, well, I think this is going to work. I really do. We're waiting on the projector to come on. Oh, come on. is coming. It's a bright light in there. Why is it not coming out here? Ah, now I know. There we go. Yippee. All right, ignore that. Here we go. Let's talk about the cranial meninges. The cranial meninges are the layers of protection around the brain. There's 3 of them, and you need to know these. We actually you will see these on your brain models, or not on the brain models so much, but you will see them on your brains. The sheep brains. You'll also see them on, you remember, probably saw them on the spinal cord when we did spinal cord last at the end of 231. So, the dura mater is the outer layer. It's the toughest one, the tough mother. I always think of Columbia Sportswear whenever I say that, because the old tough mother that runs Columbia Sportswear. Maybe y'all never watch TV. The dura is really tough. And it has really 2 layers. One is the outermost layer is the endosteal layer. Now, endosteal, you probably get the hint, that's the part that's actually affixed to the inside of the skull, the inside of the bone, okay, is the endosteal layer. And the inner layer is the inner fibrous layer is the tougher one. It just looks like a piece of thin paper, but you can see it quite easily, and it's pretty tough. The venous sinuses run between those 2 layers of the dura mater. The next layer inside is the arachnoid spiders. It looks kind of spidery, and the subarachnoid space is obviously between the arachnoid and the pia. The pia is attached to the surface of the brain itself. You can't really pull the pia off of your brain. So when you get down to the surface of the brain, you just have to take my word for it that layer you're looking at is the pia mater. It's just right there attached. The pia is the only one that goes down into all the little ridges and valleys on the brain as well. The dural folds. The dura has several folds in it that are really kind of cool. And there's some great diagrams in the book, and I will try to illustrate those for you here too. But the dural folds fold down between parts of the brain in order to protect the brain even more. We'll get to them in a minute. The most important dural folds are the falx cerebri. Falx means, what does falx mean? Y'all heard that before? What? The fault?
Curving.
Curving or sickle shape.
Yeah, it's in the middle. It's the area that goes down in between your 2 cerebral hemispheres. So it's the crease of the dura that goes down in between the 2 sides, the right and the left cerebrum, and gives it stability there so that your brain doesn't slosh back and forth as much as it could, and so the dura helps protect things like that. There's also a falx cerebelli, and you will see these on your sheep brains. The falx cerebelli, however, is about 1/10 as impressive as the falx cerebri, and it does the same thing. It goes in the midline right here between your 2 cerebellar lobes. Now the tentorium cerebelli is kind of cool, and if you just think about the name of it, you'll remember it. It's called the tentorium cerebelli. It is the tent over the cerebellum. And remember, the cerebellum is that one back here in the back, and this is a fold of dura that extends up into the head with the cerebrum above and the cerebellum below. It forms a tent over the cerebelli. Okay? Tentorium cerebelli. Also contains the transverse sinus. We're going to get to that, those sinuses in just a minute, and I have a great video to show you about that too. So I said earlier that the folds in the dura, and between the two layers of the dura, you find the dural sinuses. Well, what does that mean? Well, the blood that goes to the brain tissue itself comes up through the carotid arteries in front, and it comes up through the vertebral arteries in back, and then it percolates up through the brain. But it doesn't go out the same way as it came in, okay? And what it does is that after it percolates through the actual brain tissue itself, it finds its way into one of these dural sinuses. And they run right, the biggest one, runs right here, just underneath the skull, between the inner layer of the dura and the outer layer of the dura, which means it looks like that. It's full of blue blood. It's full of venous blood that has already percolated through the brain itself. So if this is the outside layer of the dura right here, the endosteal layer, this is the meningeal layer of the dura right here, and between those two is where we find the blood flowing, especially here in the midline and also some around the outsides, which I'll illustrate to you in a few minutes. But that's where the blood leaves, okay? Now, when the blood leaves the brain, it works its way mostly through into the central sulcus up here, the top one, and then it works its way back here to the back of the brain, and then it comes out through the transverse sinuses, and then it kind of does a loopy-loop, and it goes down back to the heart through what vessel? Anybody know? Jugular vein. Jugular vein is the great big veins in the neck that carry blood back to the heart. So these sinuses take the blood back here. It works its way back into the middle of the head. You remember the jugular foramen when we were studying the skull? So that's where it exits the skull right there and heads down to the heart. Okay. Dural layer, arachnoid layer here, spidery looking. Inside of that, the pia layer. Here's a better drawing of those dural sinuses than maybe I was giving you with my pointing at my head. But here's the superior sagittal sinus. The superior sagittal sinus runs right underneath the skull from front to back, and it carries blood in this direction, back here to this point. That's the superior sagittal sinus. Now, the inferior sagittal sinus, sagittal, obviously, sagittal cut right down the middle. Inferior sagittal sinus runs here right at the lower edge of the falx cerebri. So that the fold of dura comes down into the brain to separate the two sides, and then it folds around, creating this sagittal sinus and goes back up. Okay? Now, the stuff, this part here is open because that's where the things like the internal capsule and the different parts of the brains that connect the two sides run back and forth through here. All right, here's the tentorium cerebelli. Here it's the tent over the cerebellum. The cerebellum lives right in here. Cerebrum all lives up here. Okay? The blood, and I'll show you in a video later, flows back through here, back through here. This is called the straight sinus. Comes back to this transverse sinus. Also comes up through the falx cerebelli here to meet to this point. When he gets to this point, he goes out and follows around the edge, following this vessel here, which is the transverse sinus, and find its way down in here. It gets down to the jugular vein and heads on back to the heart. I've got an animation. I think I'll show it at the end of this period. So let's talk about cerebrospinal fluid for a minute. Cerebrospinal fluid is the fluid that is pretty much like water. It looks like water. It acts like water. It has electrolytes in it, but they're really nothing out of the ordinary. It is just fluid that cushions the brain. It kind of allows the brain to float there in water to protect it. It also interchanges stuff with the brain. In other words, it exchanges glucose, it provides ATP and all this other stuff to the brain. The real functions of it, the main one is the cushioning effect of these structures and supporting the brain. If you didn't have cerebrospinal fluid in your brain would kind of collapse. It transports nutrients, chemical messengers, and waste products. Let me just stop for a second and say this. When I was a kid, my mom and dad took me to Washington, D.C. one time, and we went to Ford's Theater, where Abraham Lincoln was shot and assassinated by John Wilkes Booth. And I was so affected by that, because there in the museum they have the actual gun, the Derringer that he shot him with, and they have his clothes, and it's all bloody, and all this stuff was there. And as a 10 or 12-year-old, it was tough on me. Well, across the street, if you remember the story, Lincoln was shot in the theater there, and they picked him up and they carried him across the street to this boarding house because they knew he was going to die. His wound was declared mortal immediately. I mean, people with brain bullets in those days didn't ever live. So they knew he was going to die. So they took him across the street to this boarding house, and they took him upstairs up to the second floor, I believe, because that's the only place they had a double bed. And he was a really tall guy, so to get him in the bed, they had to kind of lay him catty corner across it to have him fit. And a bunch of political leaders and his wife and family, they hung around that night. He died early the next morning. And the thing I always remember from what I read about it was that all during the night, Lincoln's brain oozed out through the bullet hole. And so that by morning, he had a big pile of brain tissue on the bed. Now, you guys have seen and felt those sheep brains, and you think, well, how's that going to ooze out through a hole in the brain? Well, the thing I want you to remember is that your brain in no way resembles those sheep brains. Those sheep brains have been soaked for 3 months in formalin to get stiff like that, to keep them from rotting. Our brains are very, very fluid. They're almost like loose jello, okay? So the actual consistency of our brain tissue is loose jello. And through that loose jello, there's all these little tiny blood vessels. And it's for that reason that you should never, ever, ever shake a baby. Because if you shake a baby, it's like having a piece of Tupperware full of jello, and when you shake it good, the jello inside is going to wobble around and it's going to rip and tear. You're going to get little rips and little tears in the jello, and it's going to tear those little vessels. And that's what happens to kids' brains when they get shook. It tears those vessels, and it causes very, very terrible brain damage, blindness, and so forth. Yeah.
As you age, does it firm up, or how does it
Not a whole lot. It's a little more firm, but it's nothing like the sheep brains. Okay? I just want you to always keep that in mind, that it's really liquidy. And that's also what happens in all these football injuries that you're seeing and hearing about these days. You know, a guy, yeah, he may have on a great helmet, you know, and he, you know, it doesn't hurt him at all on the outside, you know, his skin, he doesn't even get a bruise, but he flings that brain up against somebody else, and his brain inside of his skull goes boom, boom, against the inner walls, and that's where the damage occurs. Okay? Does that make sense? Okay. So remember, President Abraham Lincoln, pile of brain in bed. Kind of a disgusting thought, isn't it? It was very traumatic for me as a 12-year-old. Okay. So, this fluid that's circulating around the brain, it's in the subarachnoid space, just below the arachnoid mater, and it's also in the ventricles. Remember the ventricles. Where does it come from? Well, it comes from these things called the choroid plexus. The choroid plexus are these very specialized ependymal cells. Remember, ependymal cells are the cells that line the inside of all the ventricles. They line the choroid plexuses for sure. They line the central canal going down the spinal cord and so forth. And they're the ones, this is the areas that make the CSF. And the fluid just comes off of them and goes out into the ventricles. Now, we have really good models over there in the lab that illustrate the choroid plexus. In fact, you can see it on your sheep brains. If you look in the floor of the lateral ventricles, you look in the floor of the third ventricle, you look in the floor of the fourth ventricle, you'll see these kind of spaghetti blood vessels in there, little tiny thing, but they're very obvious. That's what makes the CSF. That's what makes this fluid. It's also it also removes some of the waste products from the CSF, and the choroid plexus even adjusts the composition of the CSF. If it gets too osmotic, starts to get hyperosmolar, it's going to correct it. If it gets hypoosmolar, it's going to correct it, because everything about the brain has to be in good shape. So how much CSF do we make a day? Well, about 500 cc's a day. Well, think about that. 500 cc's a day. There's 24 hours in a day. So what is that? That's just like 20 cc's an hour. Yeah, 20 cc's an hour is all we make of CSF. But not only do we make 20 cc's, but we got to get rid of 20 cc's, because we always want to keep about the same amount in the system all the time. So keep that in mind. Because remember about the brain, it's in a box. The brain is in a box. You put 10 extra cc's in the box, and it causes the pressure in the brain to go up. All right, so where does it circulate? Well, from the choroid plexuses, which are on the floor of the four ventricles, it circulates all through those ventricles, and some of it goes all the way down the central canal of the spinal cord. But it also goes out of the middle of the brain, and it goes out and around the brain into the subarachnoid space. That occurs in the fourth ventricle. And there's one central hole, and there's two out lateral. I've got pictures of them somewhere along in here. Okay. So that's where it's made, is in the choroid plexuses, which is in the middle of the brain, on the floor of the ventricles. Where do we get rid of it? Well, we get rid of it up here in the superior sagittal sinus, and in that area, the arachnoid villi, which are basically extensions of the subarachnoid space that extend through the dura mater into the superior sagittal sinus. Remember, the superior sagittal sinus is venous blood. Okay, so it's in this area right here, in that middle part, where our body gets rid of the CSF. So it's picked up in these arachnoid villi, it's secreted into the bloodstream, into the superior sagittal sinus, goes into the bloodstream, goes to our kidneys, we pee it out. Okay, it just gets pumped around the body a little bit, and we can control the volume that way. These arachnoid granulations are really large clusters of villi, and they absorb the CSF. I have a video, like I said, that I want to show you of this. So here's basically the idea of the CSF flow. It's formed in the choroid plexuses. Here is what it kind of looks like. Little blood vessels on the floor of the lateral ventricles, on the floor of the third ventricle, and the fourth ventricle. So all four of the ventricles have choroid plexus in it. That's where the fluid is made. The fluid then circulates all around the brain. Well, no, correction. Let's say it fills the ventricles, comes back here. And then here in the fourth ventricle, it goes out of the ventricle, the center of the brain, and goes out and surrounds the brain through these three holes. One is called, the two lateral ones are called the lateral apertures, and then there's a median aperture as well. So there's a total of 3 of them. And they allow the fluid that's inside the middle of the brain system, so to speak, to come out and go out here and go around the outside of the brain and into the subarachnoid space all the way around. Now remember, the subarachnoid space is the space down here that you stick a needle in to do a spinal tap. Remember that? So that's the easiest place to get at CSF, is down there in a lumbar puncture. As it circulates around the outside of the spinal cord and the outside of the brain, that's where it's picked up here in these arachnoid granulations and in these little villi things that are illustrated right here that sort of stick up there into this superior sagittal sinus, and that's where the fluid from around the brain goes out into the bloodstream and is taken away. Make sense? A bigger picture, I think, of the same thing. But be sure when you're looking at your sheep brains that you look for the choroid plexuses, these little, they almost look like little tiny varicose veins, little clusters of veins on the floor down there. Once again, what's this called? Tentorium cerebelli, right? That's the sinus back there that's running through the tentorium cerebelli. Sure. All right, I'm going to show those 2 videos at the end. The reason is that I don't want to goof up my recording that we've got going on here. Let's talk about the blood supply to the brain. Now, this is one of those areas that bugs me and probably bugs you. And you know, I used to do a lot of vascular surgery. So I've always thought, and Melissa will always tell you that when you say, well, what's Dr. Sessions going to ask? She'll always say, well, he's probably going to ask you more about the blood vessels than anything else. Well, I used to operate on them a lot, so they kind of come to the top of my head when I'm writing these tests out. But anyway, what is the blood supply to the brain? Well, we have 4 blood vessels that carry blood to our brain. The 2 of them are the carotid arteries here in front. You put your hand right there, just medial to your sternocleidomastoid. Push it in there. You can feel it pumping away. Now, don't push too hard; you might pass out. But that's your carotid artery. Okay. Now, your carotids carry most of the blood to your brain, but we do have 2 more blood vessels that come up the back here. They actually come off of the blood vessels that go to the arm here off the chest. Before the brachial, in the chest. Subclavian. Yeah, subclavian. They come off up in here and go up the back. And then as the vertebrals go up to the brain, they do a really weird thing. And you guys remember this, I'll bet you. You remember those cervical vertebrae? Remember how they had that little extra hole out to the side on either side? You remember what ran up through those? The vertebral arteries. So those vertebral arteries that we're talking about that come off of your subclavians here, come up the back of your neck, they go up through those 7 holes laterally, the transverse foramen they're called, in the cervical vertebrae to get up to the back of the brain. They go up through the foramen magnum, they go into the back of the head on the bottom side of the brain, and so basically what you have is 2 blood vessels that come up, and I should draw this farther down on the board. So you have 2 come up like this, and these are the vertebral arteries, and they join together here in the back of the brain, alongside just under the brain stem, and they form the basilar artery, the basilar artery. And then the basilar artery comes up here and it enters what's called the circle of Willis. Now, we're told not to name things after eponyms anymore, you know, because that's not very scientific. But I can tell you that's what everybody still calls it. This is called the circle of Willis. And I think there may be what's the other name for it? The inferior cerebral anastomotic circle or something like that. Anyway, off of the circle of Willis comes off posterior cerebral and a middle cerebral you might have to flip over to the chapter on blood vessels. And then there's a one up here called the anterior cerebral. And coming in right here, so these are all going out, okay, from this circle. But coming in, there's one more up here. I think it comes in right here. It's pretty big. This is where the carotid artery comes in. Okay, so this is the one that comes down the front of the neck. It carries blood up here. This is the carotid artery. Now, why is this important? What's so protective about this? Well, it's nice that we have 4 arteries carrying blood to our brain. Because if one of them got completely cut off or blocked off, we probably still have enough blood going to our brain. And interesting story is we used to have people walk into our office about every 4 months. We'd have one come in, and we had an ultrasound machine in our office. We scanned carotid arteries for narrowings and for problems that led to strokes. And every so often a patient would walk in and they had one of their carotid arteries was completely occluded, completely blocked off. And they didn't even know it. And what had happened is arteriosclerosis had built up and built up and built up. It got narrower and narrower and narrower, and then it just clotted off. And because of the fact that they had one on the other side, and because of the fact that they had 2 vertebrals back there, they didn't have any problem with it. And there was nothing we could do about it either, by the way. Once it was clotted off, we wouldn't try to go in and clean it out or anything, because that was fraught with more problems than it was worth. So that's why we have the extra blood supply to the brain. It's a good thing. Okay. So most of the artery comes up through either the internal carotid arteries or the vertebral arteries in back. It's removed from the dural sinuses, goes down through the jugular veins and back to the heart. Here's a nice picture of that. This is from that later chapter. This is the artery I was trying to name here, the subclavian artery, because this is where, right here, is where those vertebral arteries come up. See, they come off right here in the neck and the chest, actually, and they come up and they run through those transverse foramens in the cervical vertebrae. See that? Isn't that cool? Go up there and feed into the back. Become the basilar artery and then join the circle up there. Here's the other one, the carotid artery. The common carotid comes off here, comes up, it splits in the neck. The external carotid goes up to your face and feeds your muscles and your tongue and your skin, all that stuff. And the common carotid artery goes straight up to the brain. It has no more branches at all until it gets to the circle of Willis. Here's what the bottom of the brain looks like. If you're looking at the bottom, this is what I was trying to draw you over there. Here's those 2 vertebrals here and here coming up. They merge to form the basilar artery, and then this is the circle of Willis here, so to speak. And you notice there's a posterior cerebral artery, there is a middle cerebral artery, and there are anterior cerebral arteries that all supply the brain. And also, you have this carotid artery coming up, which is drawn as right here. It's cut where the carotid artery came up and supplied blood to this whole system as well. Now most folks, if you've ever had a grandma that's had a stroke, she's probably had a stroke because she flipped off a little piece of plaque or cholesterol and got stuck here in the middle cerebral artery. The middle cerebral artery obviously goes to this main part of your brain out here on the side, And if indeed you have a big piece that blocks off a big artery like the middle cerebral, they're probably going to be paralyzed and numb on the other side of their body. Okay? So remember that the right side of your brain controls the left side of your body. So if you take a piece of clot or cholesterol to one side, it's going to cause symptoms on the other side. Here's the venous, the blood leaving. It's collecting up in that superior sagittal sinus. It's coming back here. It takes these transverse sinuses out from the back of your head back here. It goes out. Does this these sinuses here? What I thought we used to call this the sigmoid sinus, but anyway, it heads over here to the jugular vein and heads down that way. Here's the jugular. Here, coming down to your heart. Okay, cerebrovascular disease. I got ahead of myself a little bit there, but any disorder that screws up the blood supply to your brain is called cerebrovascular disease. And anytime you have a piece of brain that doesn't get blood flow, it dies generally. If the blood flow is reestablished in just a few minutes, you may recover. We call that a TIA, a transient ischemic attack. Grandma may have had her arm go dead, and 5 minutes later, she could use her arm again. That's a TIA. That's a very serious symptom, by the way, because if you have one TIA, you're probably going to have another one, and if you have another one, it might be a full-blown stroke the next time. So TIAs are really important. There's two basic types of CVAs. One is like I've just mentioned, with a little piece of clot or a little piece of cholesterol going up and blocking an artery off. The other kind, and really the more serious kind, is when one of the little blood vessels way up in your brain here blows up and breaks and bleeds out into your brain. Now, from what we've talked about today, you can kind of figure that if you have an artery that is bleeding into your head, it's like taking a fire hose and shooting it into Jell-O, okay? And it just keeps bleeding. There's nothing there to stop it. The Jell-O just moves out of the way, and it really screws up that piece of brain. And so people that have an intracerebral bleed have a very high mortality. That's how my mother died a few years ago. She had an intracerebral bleed into her brain, and she did live about a month, but it was not pretty. So, two kinds are a clot and a blown out vessel that lead to most all strokes. Okay, I've talked a little bit about this blood-brain barrier stuff, and I know it sounds kind of like hocus pocus to you, but this is what isolates the neural tissue from the general circulation so that the brain itself is bathed in blood that is in much better shape than the blood that's circulating through all the rest of our body, because we need it to be perfect. It's formed by this network of tight junctions where the cells bond together tightly between the endothelial cells of the capillaries that are in the central nervous system. It controls the flow of lipid-soluble compounds, and two of those that are really important, of course, are oxygen, breathe in the good gas, and CO2, breathe out the bad gas. So we keep the level of oxygen and carbon dioxide in our brains very, very stable so that we can think right. It also controls the levels of steroids, prostaglandins, almost all the drugs that we take. They may have one concentration out in your bloodstream, but up inside of your brain, it might be quite different. So, what controls this? Well, the astrocytes do. What are astrocytes? Remember them? What are they? They're neuroglia? That's very good. What do they do? I'll look that up. This is one of the things they do right here.
provide structural support, regulate ion, nutrient, and dissolved gas concentrations, absorb and recycle neurotransmitters, form scar tissue after injury.
Wonderful. Okay. So, what about this blood CSF barrier? You know, one thing is to be in the blood, and for the blood going into the brain to be controlled by the blood-brain barrier, but we also have to control what gets out of the blood and gets into the CSF, right? And that happens in the choroid plexus, and that's from these special ependymal cells that are in the choroid plexus, so that only the stuff that we want to get into the CSF gets into the CSF. Because we don't want to have any bad stuff floating around in our CSF, which then affects our brain. Okay? There's 4 places where there's a break in the blood-brain barrier. Some of it's in the hypothalamus, and this will make more sense to you when we get to really start studying the endocrinology and study the makeup of the hypothalamus, because a lot of the hormones that we excrete are controlled by releasing factors that are in the hypothalamus that then travel to the pituitary gland and then cause different effects throughout the body, especially all of the sex hormones, all of the thyroid hormones, growth hormones, and so forth. And in the posterior lobe of the pituitary, we have 2 hormones made, ADH. What's ADH? Anti-diuretic hormone. And oxytocin. What does oxytocin do?
That's the bonding hormone, isn't it?
It's what makes mothers willing to hold children and let them breastfeed. Yeah, it allows the drop down of the milk for breastfeeding, and it also causes ladies to go into labor at the end of their pregnancy. Oxytocin. Pineal gland. Pineal gland is another place where there's a break in the blood-brain barrier. We'll talk a little bit about it. And this choroid plexus is sort of a break, but it works very hard to keep it controlled. Okay. I think I've said most of this that I want to say, just saying that the brain is, the meninges stabilize the brain. The CSF protects against sudden movements and cushions it because it's floating in water. The CSF, the fluid, also carries things to the brain, providing nutrients and removing some wastes. And then we've talked about this blood-brain barrier and this blood-CSF barrier. And basically their job is to very specifically, selectively isolate the brain from chemicals in the blood that might screw up our brain function. And it doesn't take much to screw up our brain function. Okay. Talk about some parts of the brain again, kind of a review really. The medulla oblongata is the main connecting point between the brain and the spinal cord. It's at the lower end of the brain. It coordinates these complex autonomic reflexes. In other words, it controls your pulse, it controls your breathing, and so forth. I think that's about all you need to know about it. It has several nuclei. What is a nucleus in the brain? A nucleus is just a collection of cell bodies that controls something, or that does one function. Don't be confused by this word nucleus. Nuclei are collections of brain cells in the brain that control one particular function. And so usually when you're looking at the slice of the brain, there'll be several nuclei you'll see there. They're different colors. They're dark usually as opposed to white. The medulla also controls motor nuclei of the cranial nerves. We'll talk about cranial nerves here a little later. And it functions as a relay station to carry sensory and motor pathways up to the brain. Here's the medulla oblongata down here. Several of the cranial nerves come off of it. What's a cranial nerve? Cranial nerve is a nerve that comes directly out of the brain. That's all it is. We have 12 of them. We number them using Roman numerals 1 through 12. Okay? You'll need to know the names of the 12 cranial nerves, and you'll need to know basically what they do, okay? Because it's testing for cranial nerve function is so interesting. If there's a problem with one of your cranial nerves or with the area of the brain that controls that cranial nerve, it's usually pretty visible. Like, you know, I can't open my eye, or I got the side of my mouth is sagging, or I can't hear out of this ear, or I can't see out of the bottom half of this eye. All of those things are those kind of symptoms are controlled by the cranial nerve. So we'll go into that. You do not need to know all the cranial nerves as they come out of the brain. I usually only hold you to 3 of them.
Just the sensory ones? Is it just the sensory ones?
Number 1 is the olfactory nerve, and it's a big nerve that doesn't even show up on this picture, but it's the nerve of your smell. And number 2 is this nerve here, which is called the optic nerve, which is the one you see with optic tract. They hook together here and go back to your brain to carry your vision. And the only other one I think you can ever figure out is number 5 right here. Number 5 is the trigeminal nerve. We'll get to it. But the only reason you can tell it is because it's by far and away the biggest of the cranial nerves. It's just a lot bigger than the others. You'll see it on your brains. Melissa pulled out the human brain. We have a human brain over there in a bucket. Did y'all see that? Yeah, that was a really, really terrible 231 student. That was a joke. Okay. I don't like to, I just want to mention these. Please don't feel like you're going to get hammered on this stuff. The different areas of the different spots, which we can call autonomic nuclei. Nucleus, remember, is a collection of brain cells that does one thing. Reticular formation is one of them. The medulla also has the cardiovascular centers in there that make your heart beat and control your heart rate, and respiratory centers that make you breathe in and make you breathe out. Now that may not seem all that complicated, but the fact is it's pretty complicated because you start to breathe in. At some point you got to figure out when to quit breathing in, right? And so it does that. And then it needs to know when to quit breathing out. That's what happens in the respiratory rhythmicity centers. I'm going to skip that. We're going to I'll go over the cranial nerves with you, probably over in a lecture. Okay. The pons, as I told you, really only does one thing, and that is it connects the cerebellum with the spinal cord and the other areas of the brain. It's basically 2 big sets of fibers that kind of wrap around the brainstem and go right into the cerebellum that allow you to be coordinated and coordinate all of this information. Now, some of you didn't have me before, and let me just illustrate what that means. If we talked about what kind of fibers needed to have the highest speed so that they could conduct the information to the brain faster. You remember it was the bigger fibers. It was the myelinated fibers that carried the fast signals. And the place we needed those the most was in our muscles and in our tendons in order to keep our muscles coordinated. And so if you just watch me, just think about this for a second. If I stand on one leg, okay, and you'll probably notice my fingers are moving a little bit. If you can see my knee, you'll see that my knee is moving around a fair amount. Now, why is that? Why? I mean, other than the fact that I'm kind of a spazzo and maybe I'm not strong enough to stand on one leg, but why is that?
Trying to maintain your balance?
I am trying to maintain my balance, yeah, and how do I do that? How do I maintain balance?
Well, it's correcting the movement. Your brain is sending signals.
That's
pawns,
right.
I guess right there.
So I'm getting all these signals up there that's saying, hey, Sessions, you need to move a little bit to the right. You need to move a little bit to the left or you're going to lose your balance, right? And the other thing is, you remember that when we were talking about muscle function, and we talked about those different groups. These muscles will contract for a second, and then when they start to get fatigued, another motor unit, we call them motor units, then another one would fire off, and then another one would fire off and let the first one relax, things like that. Well, all of that is going on all the time, and all those signals are being sent up to my brain saying, oh, that muscle fiber is tired. We need to let that one relax. And then, oh, we're leaning a little bit too much to the right or left. Well, all of those signals are going back and forth thousands of times a second, okay? And that's all it's coming up the spinal cord, it's going through the medulla, it's getting to the pons, and the pons is then sending it into the cerebellum, and the cerebellum, that's all it does, is maintain our coordination. Our muscle and tendon coordination is managed in the cerebellum. I don't hold you to this apneustic. Apneustic means not breathing. Pneumotaxic is breathing. Just know that those centers are in the pons, and that's it. Here's the medulla, and I don't hold you to any of this. All of these, you ought to know what the olives look like, because they're the one thing that can really help you orient where you are on the brainstem. This big green thing here is the olive, but don't know what it does or anything, that doesn't matter. Cardiovascular center, solitary nucleus, nucleus cuneatus, gracilis, reticular formation. Don't worry about those. Just know that this whole thing here is the pons. The pons is taking all of this information and sending it into the cerebellum to allow you to be coordinated. What are the functions? Basically adjusting the postural muscles. It also does fine-tunes your conscious and subconscious movements. You guys have seen people with
Parkinson's?
yeah. Okay, those are subconscious movements that are controlled. There's a chemical out of balance called dopamine, L-dopa. And who's that guy that's an actor that has that?
Michael J. Fox.
Michael J. Fox, yeah. And Michael J. Fox is still very functional, and he takes his medicines on time. The problem with L-dopa is you have to take it like every 4 hours. We haven't been able to come up with a long-acting pill that you can just take 1 a day or 2 a day. And you got to take it right on schedule in order to keep your dopa level just right. Well, he does that. The other guy who has the same disease that doesn't take his medicine on time is Muhammad Ali. Muhammad Ali has had Parkinson's for many, many years, and half the time he's either out of it or he's stiff up and can hardly move if you watched him. He just has a hard time taking it right. You'd think he could get somebody to just follow him along and say, okay, hey, Muhammad, here, take the pill. Doesn't work. The cerebellum itself, a lot of Purkinje cells. You need to know what these look like under the microscope. The cerebellum is really pretty because it has this thing called the arbor vitae, which is a highly branched set of white matter inside of the cerebellum that looks like a leaf. Okay, arbor vitae means a leaf. Arbor is tree, right? Vitae, vital parts of the tree, the leaves. So that's, I think that's about all you need to know about it, and I don't need to know this stuff. What is ataxia? That's a disorder of the cerebellum. I'll try later to. You guys have all been ataxic at some time in your life if you had too much to drink, and that's why when the policemen stop you, they make you do this kind of stuff, and they make you look right at them. And they make you look out to the right, and they make you look out to the left, because what they're looking for is nystagmus, which is rapid movement of your eyes. It's out of your control. And so you may look out there, but then your eyes start bouncing around. And if you've got nystagmus, you're going to the jail. And they're going to do a blood test on you, and you're probably going to get hammered for $20,000 worth of tickets for driving drunk. luckily, it's temporary for most people. And you've seen people that have cerebellar problems. They can't walk smoothly or do much of anything smoothly. Cerebellum looks like this. They call these folia, these little layers and nice little streaks across here. This is the left hemisphere. This is the right hemisphere. There's this little thing down the middle called the vermis. I'm not sure why it's called that, but it's also called the vermiform lobe, I guess, down the middle of it. The cerebellum is kind of cool looking. If you slice it down the middle, you see this thing here, and this is the arborvitae, thing that looks like the leaf, and looks like this as well. You'll see that on your brains. Some of you cut them the other day. Did you see that? Yeah. That's pretty much, you can see that there's a lot of things named on here, but I don't really think that's important. You just should know that the cortex of the cerebellum is on the outside, same as it is in the cerebral cortex is on the outside. The white matter is on the inside, down here in the form of this arborvitae thing. Notice that the cerebellum comes right off of the fourth ventricle. So if you can identify where the cerebellum is, you know where the fourth ventricle is. The fourth ventricle is important because it has a choroid plexus in it. And it also has those apertures where the fluid gets out of the inside of the brain and gets out to the outside. Somewhere along in there and there. I'm going to stop here on the slide that says diencephalon, and we'll take that up next time. But was there a class that comes in here after us? Well, anyway. I'm going to take a few more minutes to talk here in lab, in lecture rather. Let me stop this recording if I haven't already done it.